Today we will be discussing the module about the principles of hemodialysis machines as a part of the paper on medical biophysics. Now, what exactly is the process of hemodialysis? Now, the picture that you see in front of you is basically of hemodialysis machines. You see a lot of knobs around there, you see a lot of connections. Oh my god, it seems to be so complicated. Now, the dialysis process is not that complicated. So now today's module would be telling you more about the process of hemodialysis and how exactly these machines work. So the learning objectives of today's modules are to define the process of hemodialysis and to know the indications of initiation of hemodialysis. To understand the physiological principles which are applicable to the process of dialysis, and to understand the functioning and the components of different parts of the hemodialysis equipment that you just saw in the previous slide. What is chronic kidney disease? Kidney is a very vital organ of our body, performing important functions of excretion of waste products from your body. Along with that, it also maintains water and electrolyte homeostasis. Additionally, kidneys synthesize certain hormones as well as certain metabolic functions which are very vital for the body's normal functioning. Now, once your kidneys are affected, what happens is all these functions are hampered. What we are facing these days is that kidney disease has become the new non-communicable disease epidemic. Its prevalence and incidence is rapidly rising. Now, kidney disease is a very chronic and debilitating condition because there is no permanent treatment and it requires continuous process of removal of waste products, which ends up with huge amount of morbidity and mortality in the patients. The only form of treatment that is now available is what we call as the renal replacement therapy. Renal replacement therapy basically involves two modalities. One is what we call as the process of dialysis and the other is what we call as the process of renal transplantation. Renal transplantation is the most preferred treatment modality for chronic kidney disease patients, but because of feasibility issues and the availability of donors for kidneys, this process is not so widely used. So what we have is the process of dialysis. Now, dialysis basically takes care of the first two functions that we discussed of the kidney, that is the removal of waste products from the body as well as maintenance of electrolyte and water balance. Dialysis can be of two types: peritoneal dialysis, where the dialysis is done across the peritoneal membrane of the patient, and the other process is what we call as hemodialysis. Amongst them, hemodialysis seems to be more effective form of treatment because it ensures complete removal of waste products along with lesser complication. So what exactly is hemodialysis? Hemodialysis basically involves a process of exchange of waste products and electrolytes across a semi-permeable membrane between the patient's blood and an artificial electrolyte solution which is synthesized with a similar composition to normal extracellular fluid. So, in whom do you initiate the process of hemodialysis? Now, it can be used in cases of acute renal failure to tide over conditions which are life-threatening for the patient, but it is the preferred modality of treatment for all chronic kidney disease patients who end up with a condition what we call as end-stage renal disease. End-stage renal disease, or ESRD as it is called, basically means that the estimated GFR of the patient is less than 15 ml per minute for non-diabetics and 10 ml per minute for diabetics. Apart from this, any chronic kidney disease patients who has life-threatening complications, for him also we can initiate the process of hemodialysis. Now, these complications principally involves uremia. Uremia means excessive accumulation of urea and creatinine, leading to crossing across the blood-brain barrier, leading to altered mental status as well as nausea, vomiting and altered sleep pattern in the patient. Or second condition is where there is excessive volume overload because the fluid homeostasis is being affected, which can end up with cardiac failure. In such cases also, we can initiate the process of hemodialysis. Apart from this, in any patient where we feel that the existing treatment is not leading to an improvement and the patient is worsening, his renal functions are going down within 24 hours, for that patient also you can initiate the process of hemodialysis. So this brings us back to what we had already discussed about what exactly is the hemodialysis and what is the basic principles which are involved in this process of hemodialysis. As you observe in this particular picture, you see that there are two compartments. One is the blood compartment, which is shown as a grey color domain, and the other is the dialysate fluid compartment, and they are separated by what we call as the semi-permeable membrane. Semi-permeable membrane essentially means a membrane which will allow few molecules to pass through it while not allowing the other molecules to pass through it. The important thing over here is that the blood and dialysate fluid are flowing in the opposite direction. With this concept, we would be going on and discussing why these opposite direction is important, what is the role of a semi-permeable membrane, what is the importance of a dialysate fluid in the further slides. To understand this process of hemodialysis, we need to understand certain principles of physics, like what exactly is a solution, what is the process of diffusion, what is ultrafiltration, and the understanding about the semi-permeable membrane. To begin with, we would be discussing about the first physical concept which is related to hemodialysis, that is the concept of solutions. As all of us already know, solution is nothing but a mixture of a solvent and a solute. Now, dialysate fluid is also a solution which is being artificially prepared. Important thing over here is that the dialysate solution should be similar to the extracellular fluid concentration, and this dialysate solution concentration basically decides the direction of the process of exchange across the semi-permeable membrane and also how effective this process of exchange is. As you can see in this particular picture, here what we see is the semi-permeable membrane separating the two compartments of blood and the dialysate solution. Now, because the dialysate solution does not have the waste products which are present in the patient's blood, this ensures complete removal of the waste products from the patient's blood into the dialysate solution, as is depicted by the arrow. While if you see the upper panel, the electrolytes are more or less similar in both the compartments. Therefore, there is no net movement of electrolytes across the two compartments. Now, this is very important because we only want the waste products to move from the patient's blood to the dialysate solution, but at the same time we want to ensure that there is no electrolyte loss across the patient's blood, otherwise the patient will end up with what we call as electrolyte imbalance. There's another important thing that the dialysate solution is used for. The composition of this solution can be individualized to the patient's requirement. That means if I have a patient with hyperkalemia, that means there is an excess of potassium being accumulated in the patient's blood sample, I would create the dialysate solution having low potassium concentration. This would ensure that there is a movement of potassium from the patient's blood sample to the dialysate solution and helps in maintaining or restoring his electrolyte balance. The composition of a normal dialysis solution is very similar to what we've already discussed, the human extracellular fluid compartment. Important electrolytes like sodium, potassium, calcium, magnesium along with glucose have to be essentially ensured. That means the concentration has to match the ECF in the patient's blood sample. Only in special situations where electrolyte imbalance in the patient, this concentration might be changed according to the patient's requirement. Now we'll discuss about the second concept, which is very important for understanding the process of hemodialysis, which is the semi-permeable membrane. Now, as the name suggests, This membrane is permeable to certain substances while impermeable to some other substances. For hemodialysis, the membrane that we use is essentially permeable to small molecules like water and waste products like urea and creatinine, ensuring effective removal of these across the semi-permeable membrane, while it is impermeable to large molecules like proteins and blood cells, thus ensuring that the patient does not lose his proteins and blood cells across the process of hemodialysis. Now, newer membranes have a selective permeability to middle molecules also, which can pass across them, but they will move slowly as compared to small molecules because the movement is also dependent upon the size of the molecule. Now we will be discussing about the hemodialysis membranes which are being used for the process of hemodialysis. The membranes have two essential characteristics which we have to keep in mind. The first is what we call as flux and the second is what we call as biocompatibility. Flux basically tells you the molecules that the membrane allows to pass across it. Flux is determined by the pore size of the membrane. That means a membrane which has got small pores will have a low flux. A membrane which has pores of larger size would allow for high flux. So the membrane, for example, cellulose is low flux because it only allows small molecules like urea and creatinine to be filtered across it. While if you compare the newer membranes that we are using these days like modified cellulose and the synthetic membranes, they are high flux membranes because their pore size are bigger and they also allow the middle molecules to pass through them. An example of middle molecule is beta-2 microglobulin, which is normally filtered by the kidneys. But in renal failure, what would happen? This molecule will start accumulating in the patient's blood sample and can end up with a lethal disease called amyloidosis. Hence, middle molecules also need to be filtered, which is being taken care by the newer membranes we are using for hemodialysis. The second property that we will be discussing is the property of biocompatibility. Now, as the name suggests, biocompatibility means how compatible the membrane is with the biological fluid. Now, just imagine we are taking out the patient's blood sample and exposing it to a semi-permeable membrane, and hence it can lead to certain reactions because we are exposing a foreign material. Now, these reactions include decrease in the leukocyte count and complement activation. The older membranes that we used to use, like cellulose, were known to lead to immense immune reactions in the patient, while the newer membranes that we are using are more biocompatible and lead to lesser reactions. So we prefer the newer synthetic membranes for the process of hemodialysis because they are high flux membrane and they are biocompatible. Now we will be discussing about the third concept that we need to understand for the process of hemodialysis, that is the process of diffusion. As the figure in front of you depicts, there are two compartments seen, one with a higher concentration and the other with a lower concentration. So diffusion is nothing but the movement of molecules from the area of high concentration to an area of low concentration till an equilibrium is reached. So as you see the panel B of this picture, what you observe is the two compartments have reached an equilibrium and the process of diffusion has stopped. So what are the factors which affect this process of diffusion? Now as we see, this process of diffusion is determined by two things. One is the nature of solution we are using and the other is the nature of membrane. We will be discussing about both of them in detail, going on to how these two things affect the process of diffusion. So if you look at the nature of solution, there are three things that we need to remember. One is the concentration gradient, the other is what is the molecular weight of the solutes present in the solution, and third is the temperature of solution. If you look at the concentration gradient, we need to understand, like we had previously discussed, if higher the concentration gradient across the membrane, faster will be the process of diffusion. Second is the molecular weight of solutes. Like we discussed, small molecules like urea and creatinine have a faster diffusion because they have a small size. While if you look at molecules like beta 2 microglobulin, which are larger in size, they have a lesser rate of diffusion. The third thing that is very important for the process of diffusion is temperature, because temperature determines the movement of molecules. If the temperature is high, the molecules are more motile, there is a higher chances of them to diffuse across the membrane. But we need to ensure some things, because this is a biological process that we are discussing about. If we heat the solution too much, what will happen? It will become incompatible with the patient's blood sample, because we cannot evaporate the blood cells out of and denature the proteins. Hence, the temperature of the dialysate solution has to be warm, but at the same time matching to the body temperature of the patient. Now we will be discussing about the second component that basically affects the process of diffusion, which is the nature of membrane. Now, membrane has got three things that we have to consider. One is membrane permeability, the second is the surface area of the membrane, and the third is the flow geometry. To begin with, the membrane permeability determines the size of molecules which will be allowed to diffuse across the membrane, like we have already discussed, and more permeable the membrane is, obviously faster will be the rate of diffusion. The second thing is the surface area. Like we know, if we expose something to a higher surface area, what would happen? It will provide more area for the process of diffusion to occur, and hence the process of diffusion would be faster. The third thing is the flow geometry. If you remember the initial diagram that we saw that the blood and the dialysate solution are flowing in opposite direction. Now, why this opposite direction is very important? Because it ensures that the concentration gradient is maintained throughout the surface area of the membrane, and the process of filtration can occur effectively and completely. Now, we'll discuss about the last concept, which is very important for the process of hemodialysis, which is what we call as the ultrafiltration. Now, ultrafiltration terminology means movement of fluid through a semi-permeable membrane as a result of hydraulic pressure gradient. So, if you see at this particular picture, what you see is at one end you are externally applying pressure. This pressure is basically allowing faster movement of molecules from one compartment to the other. Now, ultrafiltration is this physical process which we use in the process of dialysis, wherein we use a pump to generate this extra pressure and allow for diffusion across the semi-permeable membrane. So, the basic principles of hemodialysis machines that we would be discussing now is that we use a warm dialysate solution, number one. Number two is you have to control the cycling of blood from the patients to the dialyzer, which is where the exchange process will occur, and finally back to the patient. Third is that you have to control important parameters like temperature, flow, pressure while doing this particular process. Then you have to detect any abnormal thing that happens during this process. For example, if there is an air present in the blood sample, that means you need to immediately stop the process of hemodialysis. You have to perform this process at a particular rate. That means you cannot just suck out all the blood from the patient sample and start this process. It has to be a particular predetermined rate of taking out the blood as well as pumping back into his system has to be maintained. You have to measure the ultrafiltration rate, how much pressure you have to apply for this process to occur, and with all known parameters, you have an operator who has to calculate how much fluid has to be removed. That means how long the process of hemodialysis has to be done for the patient. So if you look at the schematic picture, it clearly depicts the entire process of hemodialysis and the components of the hemodialysis machines. Now just imagine the first picture that you saw in the beginning of the module, a machine with so many knobs, so many connections, and so many parameters to be monitored. It was very confusing. So now what we are going to do is, we are going to understand that complicated process and thus make it easy. So if you look at the schematic picture, you see the patient's hand from where you have a vascular access, from where you can withdraw the blood sample of the patient. While withdrawing the blood sample, you will determine the pressure, you will determine the rate, Once the blood sample is withdrawn, you have a pump which is basically pumping the blood into the dialyzer. During this process of pumping, we also heparinize the blood. Heparinize is nothing but what we are doing is preventing any blood clotting in the blood sample of the patient. Then we monitor the pressure of the blood which is going inside the dialyzer. From the dialyzer, dialyzer is the part where the exchange process will occur and the blood will be cleansed. Now this cleansed blood has to be pumped back into the patient, and thus we have a delivery system which has to monitor certain important parameters like there should be no air leak, there should be no air present, the pressure while pumping back, okay, and finally this cleansed blood is pumped back through the same vascular access of the patient. So this is what we see. So if you essentially look at this picture, it has got three compartments. You have a delivery system which principally takes the blood from the patient and delivers it back. You have a dialyzer which basically processes the blood and cleanses the blood by the process of exchange, and you have a monitoring system. So we will be discussing about all these in detail in the further slides. So to begin with, we'll start with dialyzer, which is the most important component of the process of hemodialysis. Now dialyzer is basically a device which has got that semi-permeable membrane that we discussed. We are pumping the patient's blood into it, and it already has the known electrolyte solution which we call as the dialysate solution, across which the process of exchange would occur. Now the dialysis process would involve the physical processes that we just discussed, the process of diffusion and the process of ultrafiltration. Now diffusion is known as a conductive transfer, while ultrafiltration is known as a convective transfer. So what exactly is conduction and what exactly is convection? Now conduction principally involves exchange, but it does not have any net solvent movement across, while convection involves net solvent movement across the membrane. So the principal difference between the two processes is the solvent movement which is present in the process of convection. Now why do we need both these processes for the process of exchange? Now it's important to understand that number one, these processes preferably occur for different size of molecules. That means for a small molecule, diffusion predominates. For a large molecule or a middle-sized molecule, ultrafiltration predominates. Another difference is ultrafiltration also helps us in maintaining volume homeostasis across the patient's blood sample. That means excess fluid would also be drained out by the process of ultrafiltration. Hence, the dialyzer uses both these physical processes of diffusion as well as ultrafiltration. We'll be discussing about the second component, which is the dialysis delivery systems. Now they are basically mechanical devices which basically take the patient's blood sample from the patient, pump it to the dialyzer that we just discussed, and then bring it back to the patient. Now there are two possible classifications for such dialysate delivery systems. One is based upon the number of patients they are catering to, whether it's a single patient or many patients. And the second thing is the type of circulation they are undergoing. So if you look at the first classification, what we see is you can have a delivery system which is catering only to a single patient and the other one which is catering to multiple patients. Now both have got advantages and disadvantages. Now those which is catering to multiple patients principally are more economical because you just need one system which can take care of so many of them, right? While the other end, single patient would mean more cost. If you look at the disadvantage of this system is the composition that we are pumping through cannot be individualized according to the patient's requirement. We saw an example initially that some patients can have additionally electrolyte imbalance, water overload, which is more than the accumulation of urea and creatinine. In that situation, you will have to change the composition of the dialysate solution, which can only be done for a system which is taking care of a single patient at a time. The second thing is that in collective delivery systems, the problem is if there is a source of infection or there is an air leak all these complications will be affecting all the patients who are being delivered by that particular system, which is not the case for a delivery system taking care of a single patient. With this, we'll go on to the next classification, which is based upon the circulation. Now, it could be a single pass system. That means you send the system the dialysate solution once and you bring it back, and the other is a closed circuit. That means you recirculate the fluid again and again. Now, obviously, the efficiency of a recirculating system would be less, because that would lead to lesser difference of concentration across it and hence lesser rate of diffusion. That is why the single pass system is preferred over the recirculating system. Now we will be discussing about the last part that we have to take care of, is that the monitoring systems. Now, as you can see, in this particular picture, all those panels which are basically highlighted by the red boxes are the monitoring systems that we are taking care during the process of dialysis. We have to take care of the pressure, we have to take care of the flow, we have to take care of any air leak which is present inside the system, we have to take care of temperature and many other systems. These systems form what we call as the monitoring system. So if you look at the dialysate monitoring system, it comprises basically of two broad categories: those which are monitoring at the blood circuit and those which are monitoring at the dialysate fluid circuit. So we would be discussing about all of them in detail in the further slides. If you look at the blood compartment, you have what we call as a manometer. Manometer, as we know, monitors pressure. We need to ensure that the pressure of the blood we are taking out of from the patient sample and the pressure that we are delivering to the dialyzer all needs to be monitored. We also have what we call as a blood leak detector. Just imagine the patient's blood being leaked through some pipe while the process of dialysis. This would lead to lower pressures obviously, and obviously the patient's blood would also be lost. So we need a blood leak detector also present in the circuit. The third and the most important is the air detector. Even if a small amount of air is present in the patient's blood sample, it will lead to air embolism and instant death of the patient. Hence, these parameters need to be monitored, and these parameters are basically connected to a pump. This pump has got a valve. Immediately, if these parameters overshoot and we see a signal, what happens is this pump is closed, and hence this process of hemodialysis would be immediately stopped. The second monitoring system that we discussed about is monitoring of the dialysate fluid. Like we discussed, in this we need to monitor the temperature. It should be warm, but it should not be hot. At the same time, we also have a resistive meter, and we have a flow meter and a pressure meter. Now, flow and pressure would determine how effectively the process of exchange or diffusion will occur across the hemodialysis membrane. Here also, any divergence from the set parameters would lead to an auto shutdown mode of the dialysis machine. Now, with this we will discuss about the next concept which is involved in the process of hemodialysis, which is the vascular access. Vascular access means from the site from where you would be taking out the patient's blood sample for the process of hemodialysis. Now, there are different preferred sites, but most preferred site is the radial artery, because we know radial artery is most superficial along with brachial artery, and hence you can easily create a passage for taking out blood as well as delivering it back. So we have basically two kinds of vascular access. One is what we call as a fistula, and the other is what we call as a graft, out of which the fistula is the preferred modality of vascular access. So with this, we will summarize what we have discussed in today's module. First, that hemodialysis is one of the renal replacement therapy which takes care of the first two functions of the kidney that we discussed: removal of waste products as well as water and electrolyte balance. Then we discussed about physical processes of diffusion, ultrafiltration, and the concept of semipermeable membrane in solutions, which basically form the physical basis of the process of hemodialysis. And the third and the important thing, that there are a number of factors that affect this process of exchange across the membrane, and these parameters can be controlled as well as maintained to ensure effective hemodialysis. With this, I would like to end today's module. Thank you.